Hello friends, welcome to my channel Sam Biology Class. Today we will discuss about different types of centrifugation. Before that, I request all of my friends, please subscribe my YouTube channel and also watch, like and share my videos with your friends. Centrifugation is a mechanical process that uses centrifugal force to isolate and purify components such as tissue, cells, subcellular structures, membrane vesicles and biomolecules. This technique is also used to characterize purified macromolecules with respect to their sedimentation rate, relative molecular weight, concentration, purity, and to study conformational changes in proteins. There are two types of centrifugation. First one is preparative centrifugation and second one is analytical centrifugation. Preparative centrifugation deals with the actual isolation of biological substance for subsequent biochemical investigation. In centrifugation, the sample is suspended in a liquid and then the liquid sample is centrifuged. Depending upon the nature of the medium of suspension in which separation is carried out, preparative centrifugation is divided into two different types. First one is differential centrifugation and the second one is density gradient centrifugation. In differential centrifugation, the medium of suspension is homogeneous, having no density gradient. This technique is based on difference in the sedimentation rate of particles of different size and density. We know that large and more dense particles will move at a faster rate in a centrifugal field. On the other hand, small and less dense particles will move slowly. Because of this difference, particles of different size and density will move downward at different rate through the centrifuge tube. As a result, different components present in the sample will be separated. Second one is density gradient centrifugation, in which the medium of suspension in which separation is carried out has a density gradient. In this gradient, density continuously increases from top to the bottom of the centrifuge tube. Density gradient centrifugation is of two different types. First one is regional centrifugation and the second one is isopycnic centrifugation. In regional centrifugation, the maximum density of the gradient is less than that of least dense sedimenting particle. So the density gradient is reasonably shallow. This technique is highly useful for the separation of proteins which have nearly identical density but have slightly different size. In this technique, particles of different size will move downward through the density gradient at different rate. As a result, different components will be separated into different zones. Hence, the name is regional centrifugation. In this technique, it is essential that the process of centrifugation is terminated before any of the zone has reaches the bottom of the centrifuge tube. Second one is isopycnic centrifugation, also known as sedimentation equilibrium centrifugation. In this technique, the density gradient is steep and particles are separated based on their point density. During centrifugations, particles of different density will move downward through the gradient at different rate until their buoyant density at which They do not move further and become stationary and form specific band. So in this technique, particles which have different buoyant density are separated into distinct bands. Now the question is, what is buoyant density? Buoyant density is the apparent density of an object when it is submerged within a fluid, taking into account the buoyant force acting on it. It is a measure of how tightly packed the mass of an object within a fluid influenced by the buoyant force. Other type of centrifugation is analytical centrifugation, which is used for the characterization of purified macromolecules with respect to their sedimentation rate, relative molecular weight, concentration and purity. This technique is also used to study conformational changes of different proteins. Friends, this is all about different types of centrifugation. For more updates, subscribe my YouTube channel, Sam Biology Class.